Hello. This presentation will cover the nucleic acid testing method for COVID-19. We'll start with a little background. COVID-19 is an infectious disease that was caused by a novel coronavirus. Coronaviruses are a class of enzymes which contain genetic material enveloped by protein spikes, giving the appearance of a crown or corona. This virus was first identified in late 2019 and is said to have come from Wuhan, China. From China, it has spread to many other countries. The mode of transmission is through direct and indirect contact, mainly through respiratory droplets from talking, coughing, and sneezing. There is a wide range of symptoms, such as respiratory complications, fatigue, shortness of breath, and body aches, but in some cases it can lead to pneumonia, kidney failure, and even death. No vaccine is available yet, but treatment and a vaccine are in development. Now we'll move on to testing. PCR stands for polymerase chain reaction, which detects genetic information of the virus. PCR is used to amplify DNA, so if you start with a small amount, large quantities can be produced through thermal cycling. Thermal cycling steps include denaturation, annealing, and extension steps. Denaturation takes place around 95 degrees and is used to denature double-stranded DNA. Then, the temperature lowers to around 50 degrees Celsius to allow annealing of the forward primer to its complementary part of the single-stranded DNA template. In the extension step, the DNA polymerase synthesizes a new DNA strand complementary to the DNA template strand. After the first cycle, the double-stranded DNA is obtained. It will continue. In cycle 2, the DNA double strand will denature once again to 2 single strands. Then annealing of the primer and TaqMan probe to its complementary part of the target DNA. TaqMan probe contains a fluorophore covalently attached to the 5' end of the oligonucleotide probe. Fluorescence is emitted by the fluorophore when it is excited by the cycler's light source. Also, the probe consists of a quencher at the 3' end. The close proximity of the reporter to the quencher prevents detection of its fluorescence. In the extension step, DNA polymerase synthesizes new strands when the polymerase reaches a TaqMan probe. It will cleave the probe, separating the dye from the quencher. Each cycle will have more dye release to increase fluorescence intensity. This method allows the estimation of the amount of a given sequence present in a sample. For each strand of DNA amplified, a fluorescent molecule is produced. Fluorescence quantitative PCR instruments can monitor the number of cycles, the CT value, in which the fluorescence reaches the preset threshold and is related to the concentration of viral nucleic acid. The higher the concentration of the viral nucleic acid, the smaller the CT value. This picture shows each step of the PCR testing. The sample is collected, and the nucleic acid goes through reverse transcription, then amplification, and the results are obtained. As far as instrumentation, 2 instruments can be used for this method. The first is the Applied Biosystems 7500 real-time PCR instrument, and the second is the Bio-Rad CFX96 real-time PCR instrument, along with their corresponding software manager programs. As far as the procedure, nasopharyngeal or oropharyngeal swabs are obtained from the patient and stored. This table shows the contents of a real-time PCR testing kit that's used to obtain the swabs. Once the swab is obtained, nucleic acids are isolated and purified. After purification, the PCR system is used to amplify the nucleic acid. To conduct RNA extraction, 10 microliters of a bacteriophage is added to the specimen. The first step is to lyse the sample in order to inactivate RNases and ensure the isolation of viral intact RNA. Next, buffering conditions are adjusted to provide optimum binding of the RNA to the QI membrane when the sample is loaded onto the spin column. The RNA binds to the membrane, and contaminants are efficiently washed away in 2 steps using different wash buffers. This table shows the reagents used for the PCR reaction. After the PCR reaction is ran, the PCR instrument is set up for detection. A new protocol is created first, and the thermal profile is defined. Next, the plate is set up and the run is started. After completion, the results are interpreted by comparing the measured value to an expected value and determined detection. And that's it for PCR testing method. There are advantages and disadvantages to this method. Some of the advantages include faster results are obtained when compared to culturing, and it can be used to detect pathogens that may require long cultivation periods. Some of the disadvantages include producing false positives and false negatives. False negatives can come from insufficient sample collection, amplification inhibitors in the specimen, and non-homogeneous sample distribution. False positives can come from mislabeling and contaminants. Another disadvantage of PCR is that it is limited. This should be coupled with culturing to increase efficiency as a diagnostic tool. And that's it for this presentation. Thank you.